Okay, so this is our introduction lesson, teach one another
With
problem.
a gene, right?
Yep. Mhm.
I thought that it was doing that thing again where it
Oh,
echoed.
I think we're good.
Okay. So I have a gene is a unit of heredity that is passed on from parent to offspring and determines a certain trait.
Yeah, and then the one I have is a gene is a unit of heredity that contains information for the creation of a functional product. So, um, I found I found my definition in the book, I guess.
Okay.
Yeah.
I don't do you have the book?
Yeah.
Yeah, I don't have the book.
I didn't realize it was optional to get,
Okay. Where are
but
you in Rexburg or are you
yeah. Mhm.
Okay, me too. That's cool.
Okay.
I didn't get the book. Where are you doing your internship at?
At the Student Health Center.
Oh sweet.
Yeah.
Very cool. And a chromosome set. I have one copy of each chromosome from each homologous chromosome pair found in a diploid cell.
Okay. And then the answer that I found was a chromosome set is a group of chromosomes that can be inherited paternally and maternally in sets of 23 chromosomes. And then these sets match each other, which is pretty much what you just said.
Like, I think it's important that we know that a gamete contains a chromosome set.
Yeah.
Um, so the next one, homologous chromosomes that carry the same gene information from each parent cell, and they'd be similar in size.
Yeah, that's the same answer. Pretty much the same answer that I got as well.
Okay, sweet.
Okay. And then I got sister chromatid. So I took a while trying to find this one, but the one I got was pairs of replicated chromosomes that are attached to each other at the centromere, and they're genetically identical.
Yeah, that's what I got too. So it's like, I think it's before meiosis in the S phase, it duplicates.
Let me check. Sorry, I'm just gonna look at my diagram so I can.
Yeah,
Before meiosis.
before meiosis in the S phase,
Uh-huh.
it duplicates. So there's like a chromosome, but it's in the shape of like, um, so it just looks like a chromatid, but it's called a chromosome.
Uh-huh.
In the S phase, it duplicates, so it goes from looking like that
Uh-huh.
to it just duplicates, and that's how it gets the double shape.
Okay, and then the each one is called a sister chromatid, like each side.
Mm-hmm.
Oh, okay then. All
But
right.
originally, like whenever it's not duplicated, it's not a sister chromatid. It's a chromosome.
It's just a
When it
chromosome.
is duplicated, that's called a chromosome as well. It just has two sister chromatids.
Okay then, yeah. Okay, that's where I was kind of stuck. I was kind of like, wait a minute. Like,
Confusing because you think like a chromosome, like you can't call two different things a chromosome,
a
but
chromosome. Yeah. I forgot again. Okay.
uh, what'd you get for allele?
An allele is an alternative form of a specific gene in that it's found at the same place on a chromosome.
Mm-hmm. That's what I got too.
Yeah.
And the place that it's found is called the locus,
Oh,
right?
yeah.
Okay. Homozygous occurs when either two dominant or two recessive alleles are present. Is what I got.
Yeah. Okay, mine says homozygous describes a diploid individual with a pair of matching alleles. So it's pretty much what you just said, identical alleles of a particular gene.
Okay. And heterozygous, what did the book say on that one?
It describes a diploid individual that has different alleles of the same gene.
Okay, that's basically what I got. Um, Dominant, I said the allele trait that is expressed. Is that what the book said? I didn't really like. I kind of just
Yeah.
used
So like
that. words
I
because I
think.
don't have.
Sorry.
I said I kind of just use my own words because I don't have the book, so I kind of use the PowerPoint and stuff.
Yeah.
So
Yeah.
like
I kind of
I want
switched.
to know what the book
Some
says.
of the book was a little bit like, that just went over my head. So I'm gonna go do the PowerPoint.
Yeah.
But I got pretty much the same definition. It described for dominant. It describes a phenotypic trait. It says it describes an allele that determines the phenotype in heterozygous condition.
Okay. Sounds good.
Mm-hmm.
And recessive, I said the allele trait that is hidden.
Yeah. Mm-hmm. It's the same as I got.
And genotype is the composition of alleles for a particular trait. I got that off the PowerPoint.
Oh, okay. Genetic composition of an individual, especially in terms of alleles, which include gene. So, yeah, it's the same thing. And then the observable trait.
Yeah, that's what I got too. So, like, genotype is the ratio of genes versus phenotype, which is the ratio of like the physical traits presented.
Mm-hmm.
Okay. And then so I guess we should go over our drawings now.
Yeah. Mine's just kind of like really packed. The first one.
Okay.
Do you want to show yours for the first one, and then I'll show you mine for the second one?
Sure.
There's two lines.
So in prophase, I think we're mainly dealing with the zygotine and pachytene.
Yeah. Mm-hmm.
So in the zygotine phase, the synaptonemal complex forms and it links the two homologous pairs together. In the pachytene stage, crossing over occurs, and that's random. And it kind of I thought that keeps the homologous pairs together as well before metaphase. And then in prometaphase, the nuclear membrane starts to dissemble.
Yeah. I think you need to move your book more towards the center, just because like it gets blocked a little bit. Yeah, there we go.
The nuclear membrane starts to dissemble, and they start to align on the metaphasal plate, and the homologous pairs stay together. And then in metaphase 1, they're on the metaphasal plate completely, and the pairs are still together. And there's no nuclear membrane. And then in anaphase 1, they're pulled apart. Mm-hmm. So whatever crossing over occurred, and they assemble like randomly. So when they're pulled apart, it can be in different ways depending on how the pairs aligned on the metaphasal plate. So anaphase, they're pulled apart. So in telophase 1, then instead of having homologous pairs, they only have the chromosomes that are like there's no longer homologous pairs. Does that make sense?
Yeah. Mm-hmm.
Okay. So, and you have 2 daughter cells.
Okay. Sounds good. So then I think for the next question it continues to like meiosis 2.
Yeah.
So I'll go over that part. Okay. Hopefully I'll hold this up. Let's see. So I'm not even sure what I'm holding up right now.
Good. Yeah, it's hard. I'm like looking over like.
So like for this one was for prophase 2, the nuclear membrane's breaking apart, and we're starting off with the two daughter cells that ended in the new diagram, and it goes on to prometaphase, to prometaphase 2, where the nuclear membranes it's starting to fragment, and the microtubule spindle is forming and connecting to the chromosomes in the different daughter cells, and then it just like in your, I don't know if you just like in your diagram, it goes on to metaphase 2, where again it's aligning on a metaphase plate and preparing for anaphase.
Mm-hmm.
And then at anaphase 2, that's when they're starting to separate again. So there we go.
Very nice.
And let's see. Go on to telophase. And then in telophase, this is again where it begins to create the new daughter cells.
Mm-hmm.
So it's separating this time into 4 daughter cells with half the chromosomes that it had started off with before. And then you have 4 haploid daughter cells.
Very cool. So that's like, I think that's what confused me too. Is like at the end, those are each called, like, I think it's the difference between a haploid chromosome and a diploid chromosome is like just the one squiggly line is a haploid, and then whenever it replicates and makes the 2, that's when it's a diploid, but they're both chromosomes.
Yeah. So like that's what I always have to like remember, is that it's not just because there's 4 cells, it doesn't mean that it's gonna go back to having what do you call the number of chromosomes that it started off with.
Yeah.
I mean, oh, so it's going, it's reducing again. Okay. So let's see.
Next, number 4. Let me pull that up. I am like still in bed, so no judgment.
Oh, don't worry.
I've been working on this all morning.
Yeah. I just started the other, I think this, like I'm partway through the second assignment.
Yeah, me too.
Fine genetics.
So for this one, and I could be wrong, because it helps if I draw it out, but I didn't draw it out. I just tried to like do it mentally. But for the chimpanzee, so prior to DNA replication, have you done that one?
Yeah, so it's the in chimpanzee, the diploid chromosome number is 48.
Mm-hmm.
And so, then, did you get that as your total number of chromosomes?
Yeah, prior to replication, 48.
Okay.
And this is what I was confused about. On the part is number of homologous chromosome pairs.
So I put 24. What did you put down?
I put 24 as well.
Uh-huh.
But it's prior to replication.
So prior to replication.
There's still like, so haploid, right? There's still one haploid from the mother and one haploid from the father. So that would be pairs, right?
Yeah.
Okay. So that's why I put 24.
Okay. And then did you put 48 for sister chromatids?
There's no sister chromatids yet because
because it hasn't gone on to the next stage. Okay, that's the part that was like really confusing.
Yeah.
And that's why I kept going back to the definition. I was like, I think I'm missing something going on.
So I don't think there are any sister chromatids yet.
Okay.
And then beginning of metaphase 1, there's still 38, right? Total. And then there's still 24 homologous chromosome pairs, but they're diploid now.
Okay.
Because replication has started. So there's 96 sister chromatids.
96. So I got the 96 sister chromatids. How many did you get for chromosome pairs again?
I got 24 homologous chromosome pairs. I think. That's what I got. What did you get?
I think I mixed that up. Okay.
Because
So I'm getting it now. So like, because you can now call a homologous pair of chromosomes.
so I think that it's so
Like it'll have the same number of chromosomes because a homologous pair is now, even though it was one haploid before,
now
making it more, but it's now reduced still in number because you can call a homologous chromosome just a chromosome, just like you would call one.
yeah.
Okay.
So that's what I think. That's my. But they have sister chromatids, and that's the difference. Is now that it was replicated, you have sister chromatids.
Okay. Could you go over that again? So you got total number 48.
Yeah, total number 48, and then it's still got 24 homologous chromosome pairs, except
Uh-huh.
it's a boy. And so there's 96 sister chromatids.
Okay. For the end of telophase 1, correct me if I'm wrong.
Okay.
I said that there was a total number of 48, and then homologous was 24, and then sister chromatids 48.
Okay, so at the end of telophase 1, so at telophase 1, so there's, I gotta draw it out a little bit. So before telophase 1, or at the beginning of telophase 1, there's the homologous pairs. So there are 24 homologous pairs. And then at the end, they split into two because they pulled the pairs apart.
Uh-huh.
So at the end, there's 24 homologous pairs.
So, yeah. Okay, that's what I got. So 48,
So
24.
24 total.
Uh-huh.
And then they split them apart. They split the pairs apart, so there's no more homologous pairs. So there's 0 pairs.
Oh.
Because they took them. Let me see if I can get the picture.
Oh, I think I need to go over this question. Definitely need to go over this question again.
So like there was 4. I don't know if my picture is gonna help at all, but look at yours. And at first in anaphase there's 4, right?
Mm-hmm.
And then by the end of telophase they've reduced the number to half because they pulled the pairs apart. And so now there's no more homologous pairs because they pull them apart into separate cells.
Yeah. Oh, okay then.
If that makes sense.
Uh-huh.
But and then we still have 48 sister chromatids.
Uh-huh.
Because it's still a diploid chromosome.
Okay.
And we've still got 24 chromosomes in there.
Okay.
Does that make sense?
Yeah. It's like starting to make sense to me. I think I looked at it like completely differently. Because I did like go back and forth with this question, like changing my answers on it.
A while too. And then what did you get for end of telophase 2?
So for the end of telophase 2, sorry, I'm just gonna look this up. I said that the total number of chromosomes was 24, and then 12 and 4 homologous pairs, and then 24 sister chromatids. But I'm like still wobbly with my answer.
Okay, so I got that there's 24 total too because we still have those chromosomes from meiosis 1. So in meiosis 1 we had 24. So there's still 24 chromosomes. But by telophase 2, we pulled apart the diploid chromosome.
Yeah.
So there's no more homologous pairs either. I mean, there weren't homologous pairs by the end of meiosis 1 because we pulled those apart in meiosis 1. So I got 24 total number of chromosomes, no homologous chromosome pairs, and then because we split apart the diploid chromosome, we don't have sister chromatids anymore.
Oh, okay then.
Because by the end they're haploid, so we split them apart. So I got 0 sister chromatids.
Okay then. Yeah, I'm starting to see how you got your answers, but I'm still can't figure out how I came to mine.
I would just like look at, I don't know, I just kept looking at my pictures.
Yeah.
It took a while though.
Yeah, it makes sense to me now that I'm like looking back, and then after we had gone over the first few of these. What did you get for the last part?
So after fertilization, so we're combining 2 cells again. So I got 48 chromosomes because we're taking the 2 haploids and putting them together. So there's 48 chromosomes, and it's from the mom and from the dad, the haploid. So it's like the first one prior to replication.
It went right back to how the first one was numbered, right?
Yeah, so 48, 24, and then 0 sister because they're haploid.
I'm definitely gonna review this one more, just so that it sticks. Okay. You just move on to number 5?
Sure, if you're stuck, I can try to, I know.
I understand how you got to your answers now, but for me to remember it, I need a review. Yeah.
Got it. So for number 5, I said the genotype was heterozygous 50%, homozygous recessive 50%.
Okay, so like a 2-to-2 ratio.
Yeah.
Yeah, I got the same thing.
Yeah. And then for phenotype, tall is 50%, dwarf is 50%.
Yeah.
So it's like the same thing we crossed.
Yeah.
And we were just supposed to do the phenotypes for
number 6.
For number 6, right?
Yeah. So what I got was that there's 2 constricted and green offspring. Did you get one of those?
Yeah.
And then 2 smooth and green offspring, and 6 constricted and yellow offspring, and 6 smooth and yellow offspring.
Okay, let me just check one thing.
Okay. I guess I have mine.
That's what I got too.
Yeah.
I guess that's all we had to say. What are the predicted
phenotypes? Traits, yeah.
I did the ratios, but that's what I got too. So there's yellow and smooth, yellow and constricted, green, smooth, green. All right, sweet. And 7.
I'm hoping that we have the same answer for this one.
I did too. I got 1 out of 64.
Okay, yeah, that's definitely what I got. So it was half times quarter times quarter.
Yeah, that's what I got.
Okay.
Cool.
All right. Because I did this one twice, and I was like, is the first answer right or the second answer right? Yeah.
Yeah, awesome.
All right, well, thank you.
Yeah, thanks. I guess that's all we do, and I'll just copy. Do we both need to post into the...
I don't know. I'm going to check that.
I mean, it wouldn't hurt if we both did it.
Yeah. So if you could send me the link.
Yeah, I will.
Okay.
I'll email it to you real quick.
Thank you.
Have a good day. Nice meeting you.
Nice meeting you too. Bye-bye.
Bye.